discovery process. Our research question is, can biomedical semi-supervised machine learning be implemented in the drug discovery or approval process for Huntington's disease? Now we will be talking about our background research. Our project has the potential to change the drug development process for all diseases, not just Huntington's disease. Machine learning has been utilized in the development of cancer treatments, whereas diseases like Huntington's disease have not yet been explored. Through this machine learning approach, Huntington's patients can get new and better treatments with the potential to treat the root disease and not just the symptoms. This approach also has the potential to shorten and reduce the cost of the current drug discovery process. Huntington's disease is a progressive neurological disease that is caused by extra repeats of CAG segments. This can lead to a longer Huntingtin protein, which is later cut into small toxic fragments that bind neurons together and disrupt normal neuron function in the striatum. Huntington's disease is characterized by uncontrolled movements and loss of cognition, and it can be treated by movement control and antipsychotic drugs. Some examples of FDA-approved movement control drugs are tetrabenazine and deutetrabenazine. Some antipsychotic drugs are haloperidol and risperidone. The main contrast is that antipsychotic drugs have worse side effects. Haloperidol can worsen involuntary contractions, and risperidone has more manageable side effects, but should still be used with caution. The first step of the drug discovery process is the discovery stage. In this stage, toxicity, drug metabolisms, formulation, and chemical development are explored. Next is the development stage, which contains 3 phases. Phase 1 tests the bioavailability of the drug. Phase 2 studies the dose response in patients, and Phase 3 consists of efficacy and safety studies. The preclinical and clinical developments are also conducted. Drugs are monitored while they're in the market in Phase 4. Drug development generally takes around 12 years. From the Tufts Center for Study of Drug Development, it is predicted that $2.6 is required to create a $2.6 billion is required to create a drug. Drugs have a very small success rate of one drug being successful in 5,000 trials. Machine learning uses diverse data in large volumes in order to learn and predict the output answer to an input question. Semi-supervised machine learning classifies data sets and solves classification problems when only some data sets are labeled and others are not. Machine learning can take data from current drugs for a disease to search for other applicable drugs to treat that disease or its symptoms.
We'll be moving on to our hypothesis next. First, our sub-question is, can biomedical semi-supervised machine learning help address scientists' needs for successfully identifying drugs for Huntington's? And we hypothesize that biomedical semi-supervised machine learning will be able to identify drugs for Huntington's by labeling developmental drugs and view the similarities and the differences with drugs that are already being administered.
Now let's take a look at the experimental procedures. Children learn through experiences, the brain learns through experiences, and similarly, a machine learns through experiences. However, our experiences and that of machines are different. Machines experience data, and for our experiment, we have to collect these critical data sets. The first one is FDA-approved drugs for Huntington disease, as these provide similar effective drug characteristics, teaching and aiding the machine when it itself enters the predictive stage. FDA fatal drugs for Huntington disease, as these drugs introduce similarities of ineffective drugs, teaching the machine on characteristics to look out for. Drugs treating symptoms of Huntington's, as this assists the acknowledgement of acceptable drugs not directed for the neurodegeneration. And finally, drugs for other neurodegenerative diseases and neurological disorders, because these drugs are analogous to those for Huntington's with differences in targets as well as effects. This advocates shaping the classification algorithm for predictive learning. After collecting the data, we move on to manipulating them. We do this by randomizing the data and then splitting it in a 7:3 ratio for training set to test set respectively. After splitting the data into sets, we cluster it via the divisive hierarchical clustering algorithm. This algorithm takes a top-down approach in splitting the large group of data into subgroups based on similarities. Our next phase of the experiment is teaching the machine to predict. We used the naive-Bayes tree hybrid algorithm consisting of two different algorithms. Naive-Bayes trees, which gives a binary outcome built on the probability of where a data set is located, and decision tree, which classifies based on parameters we set. Examples of some of these parameters are successful absorption in the intestine into the bloodstream, crossing the blood-brain barrier, good binding affinity with the target site, and producing the desired effect. A hybrid algorithm is implemented so that the prediction is not only a verification system on the chance of success, but also an advisable aspect on which the scientist can improve if the drug is unsuccessful. After creating the model, the teaching process begins. Once scientists believe the machine has learned, they may test it, creating a realistic effect as we're introducing a drug it has never seen before. If the machine passes the test, scientists can embrace the opportunity and improve, or implement the machine into the drug discovery and development process. If the machine fails the test, it will begin the training cycle again.
The most common strategy in combating Huntington's disease is to inhibit Huntington's aggression, lower its levels, or do a combination of both. This can be done by activating cellular mechanisms promoting the degradation of Huntington or to silence the protein's genes. Since many of the symptoms of HD are associated with the modulatory role of the glutamate receptor function and biphasic changes in the DA transmission, treatment should be tailored towards the temporal progression of receptor and neurotransmitter changes. The significance of this is that different treatment targets would be appropriate at different stages of HD progression. Therefore, our approach may yield drugs useful for various disease mechanisms and time points of symptom progression. When the machine is learning the similarities between existing drugs, it will specifically be looking at dopamine D1 and D2, adenosine A2A, or GABA receptors in the striatum. Coria, developed in HD patients, is treated with atypical antipsychotics, targeting and inhibiting dopamine D2 receptors. So therefore, when the machine is running through olanzapine, risperidone, quetiapine, and zibrasidone, which are all atypical antipsychotic drugs, it will find D2 antagonist properties and register this in its system. Coria can also be treated with DA depleting agents, such as tetrabenazine or deutetrabenazine. These drugs decrease DA content in presynaptic vesicles and inhibit vascular monoamine transporter type 2, so that when the machine runs through these drugs, it will find these similarities and log it in its system. At this point, the machine has completed step 1, early drug discovery in the drug developmental process. Once a drug is determined to have a 90% efficient rate by the machine, it is classified as a potential candidate for steps 2 and 3, the preclinical and clinical trials. These trials are critical in determining the safety and efficacy of the drug. In the preclinical research, the drug is tested on non-human subjects, in vitro and in vivo, for toxicity and pharmacokinetic information. Success in preclinical trials precede clinical trials, which focuses on human use. This process requires information like metabolite stability in biological samples. And additionally, there are 3 main phases in this trial. Phase 1 is testing the drug on healthy volunteers, then phase 2 and 3 on patient populations. From this, it moves forward for holistic FDA review and needs NDA or ANDA or BLA applications to be submitted. If the FDA grants approval, the safety of the drug will be monitored post-marketing using the FDA Adverse Event Reporting System database. If it does not show any major problems, it can be concluded that the drug the machine discovered can be classified as one that is effective in treating Huntington's disease.
Now let's move on to the dialectical considerations. It's important to note that when thinking of the machine itself, it is not capable of applying the biochemical assays on its own and would need the supervision of a trained professional. Furthermore, when calculating the binding affinity, it needs to be outsourced to an additional computer with the computational models of the drug and the target site's interactions, which in turn will feed into the machine. Another point raised is if the machine would not eliminate the drug discovery process entirely. One of the shortcomings of machine learning and drug development is its inability to predict side effects. Still, it will considerably shorten the amount of time being spent. The machine has limitations due to the fact that it's making drugs for the disease of the general population, as opposed to a more idiosyncratic approach that's meant to meet the needs of individuals, which it could pose concerns for meeting the needs of minorities. Let's look at the ramifications, or refuting. The machine is also designed with the sole purpose of finding new drugs, so it does not calculate the other parts of the drug process, such as dosages, how the drug is administered, side effects, the combination with other drugs, etc. Due to these factors, one could argue that the proposal could be held ineffective, or simply. Full transparency is of the utmost importance when designing a proposal that requires informed consent. The participants need full transparency regarding the drug ingredients, how they were discovered, as well as any and all possible side effects and risk factors. Despite the drug passing trials and gaining scientific approval, the public may be skeptical because the machine made the drugs. With this new technology, it can become a question of whether we should turn the focus to the individual needs or that of the communities. Which would be more ethically correct? Which would be more ethically feasible? In our healthcare system, many individuals are unable to afford the care that they are needed to treat their diseases. Considering the time and money being spent on this machine, it is very possible that the price for the drugs would be extremely expensive, which in turn would make it difficult for individuals of low or unstable economic status to afford the care they need. Another concern is that since many pharmaceutical companies develop drugs to treat symptoms and not the disease, they could potentially exploit patients and create a system of dependency for financial gain. Furthermore, we must consider what would happen if the machine development process results in the harm of a patient, who will be held accountable. It's important to think of any and all ramifications that could arise when working with biochemical semi-supervised machines and the drug discovery process in general.
Thank you for listening, and we're happy to take questions. Here are references.
Okay, Morgan, you can go ahead.
Wow, you guys, well done. That's such a fascinating topic, and congratulations. You should all feel really, really proud of yourselves. You have officially presented your research proposal after 2 long weeks of hard work, and your hard work totally showed. So congratulations, I'm really impressed. All right, and now we are welcoming the panelists to join you, and you can receive feedback from them.
Big wow. So I have a question for you all. This seemed to be an original, or at least semi-original research proposal. In other words, I mean, clearly the idea of machine learning and the use of machine learning in various forms of biomedical translation is a thing. But I guess my question for you is this particular approach something that you actually developed, or is this something that you've reported from existing literature and simply related to disease and drug development for Huntington's?
So I can take this question. So this has not the specific approach, but machine learning has been used to predict possible drug candidates for popular diseases such as cancer, like we mentioned in our presentation. However, this specific approach of using the divisive hierarchical clustering system and such is something that we developed on our own, as we first started out with kind of looking at the overarching theme of
Got it. You can stop right there. You actually answered my question. I don't need to know how you did it. I was just concerned about that. Have you written this up? In other words, in each of your successive parts that you presented, is there actually a script or a transcript that you have written up?
Yes.
I would really like to see that. And the reason for that is I believe that with a little bit of massaging, this represents something that could be submitted to the international literature for possible publication. So big kudos to you. Let's bring on Professor Shook. Let's bring on Dr. Wertzman, and let's bring on Ms. Thompson to talk about content.
Let me unmute myself if I'm going first.
Please.
Tremendous clarity here. The potential for AI machine learning to sort through data at a speed, right, beyond human scientific methods. I think you have sort of a general heuristics. And as Dr. Giordano has pointed out, this could be very helpful. One thing that sort of was occurring to me was, would the heuristic have to perhaps be modified depending on what stage or what method you're using to try to identify a drug? So for example, one method would be data mining across current literature. It could be applied to, right, analyzing 1 million already published articles about innumerable kinds of drugs that we already know exist, looking for correlations, looking for patterns, things that have helped with something that might help that. So data mining would be one of them. Another one would be a huge headache, as you've probably run across in literature, is bioavailability. You can come up with the perfect drug that will work, you know, in the cell you're targeting. It ain't getting past the liver. It just isn't. That's right. So you have to worry about, you know, is it even going to make it in into your target site? And AI might help with that in a different way than data mining, because here you know kind of what the liver is going to do to a particular candidate molecule. But I think your heuristic is most easily applied to just trying out millions, if not billions, of possible molecular shapes in order to have some sort of chemical affinity and efficacy with whatever target protein or, you know, genetic replication sequence or whatever. With Huntington's, right, it's, you might even target something going on in the chromosomes. Who knows about the possibilities of this? So I think this could go in several different directions, and I also appreciate your clear cognizance of you know, some serious obstacles along the way. But you're to be congratulated for ambition. Kudos.
Dr. Wertzman.
Wonderful, wonderful job, all. It's not insignificant that Dr. Giordano says that this is something that could actually be of interest in the literature. And I want to commend you all, a really ambitious topic and the clarity with which you distilled this information. You know, this was a very, very student-directed project. You know, I could point you in the right directions, but I saw you guys really teach yourselves about all of these different constructs, machine learning, Huntington's, and you presented it with the clarity that lay people could even understand. The not just the clarity, but the thoroughness with which you thought through the various aspects of the problem, from the different types of targets, the different stages of progression. I know that you considered issues related to bioavailability and crossing the blood-brain barrier and things like that. This was a very sophisticated project that you executed just very well, and I'm very, very proud of you.
Outstanding. John, Rachel, thank you. I simply just, I'm speechless, and I rarely am, but wow, great, great work. Ms. Thompson.
Please, all of you, go to medical school. The future of the world is safe if we're in your hands. Yeah, wow. To Dr. Wertzman's point, the clarity to a layperson, which I am definitely, when you gave us the topic at the beginning of the presentation, I'm looking at it thinking, what does this mean? I don't know what they're talking about. And you explained it. Your terms were clearly defined. Everything was carefully broken down in layman's terms and then built up very quickly. Halfway through the presentation, it's like, not only do I get it, but oh my gosh, I get the implications here. This is groundbreaking, at least from my non-medical background.